Uh, those are the main things. Uh, 6, stomata, leaf opening, that allows gas exchange. NADH is reduced to NADPH rather. I had NAPD there, it should have been NADP. 8, chlorophyll is the main type. A is the main type of chlorophyll. Number 9, why is chlorophyll green? It reflects green light. Wavelength best absorbed by photosystem 2 is 680. Number 11, type of electron flow is cyclic. Number 12, what happens to the oxygen in water? After the water molecule is split, it's released as a gas. 13, name of the sac-like structure in chloroplast that contains chlorophyll is thylakoid. 14, the molecule that transfers electrons from the second electron transport chain to NADP is ferredoxin. 15, the 3 phases of the Calvin cycle: carbon fixation, reduction, and then regeneration of the rubisco. Or not rubisco, but ribulose bisphosphate. 16, the 3 plant types to minimize photorespiration. 16, what is carbon dioxide reduced to in the Calvin cycle? A sugar, CH2O, GP3, any of that would be okay. 18, long distance signaling in plants and animals, hormones. 19, the 3 processes that cells receiving signals go through. You get the reception and then the transmission and response. 20, term used to describe a signaling molecule is a ligand, Ligand. Cell death designed to protect the organism, apoptosis. 21. 22, 2 common second messengers, cyclic AMP, AC, and calcium ions. 23, all of the cells of the body except reproductive cells are somatic. 24, cell number of animal cells, 2N. Would a gamete be described as haploid or diploid? Is haploid. Number 26, a lot of people missed. The embryo develops from which process? The embryo develops from mitosis. The egg and the sperm is formed by meiosis. Once fertilization occurs, you have a diploid cell, a zygote. Those zygotes multiply by mitosis. When mitosis occurs, do cells change the genetic code in any way? The answer is no. The phase of interphase where DNA is replicated is the S phase. 29, the 3 parts of the cell cycle: interphase, mitosis, cytokinesis. Once again, you got to pay attention to the terminology. The cell cycle are those 3. The 5 stages of mitosis: prophase, prometaphase, metaphase, anaphase, telophase. The process that separates 2 newly dividing daughter cells is cytokinesis. Some people put cleavage, that's okay too. 33, I gave a lot of credit for different, any name of a fiber. Some people put asters is part of it, so I gave credit. Should be basically the spindle fibers. You could put kinetochores. I gave credit for a lot there. 34, the part of the chromatids where the kinetochores attach are the centromere. That is what holds citrochromatids together. 35, the name of the phase of mitosis where the paired chromosomes line up at the equator, metaphase. 36, what do non-kinetochore microtubules do? They elongate the cell or push against each other. 37, in plants after telophase, this forms to separate the cells, cell plate. 1, And one thing I want, still, people don't understand, I was kind of disappointed in this one question and the responses. It says to explain the difference between DNA and chromosome. DNA is the genetic code, the sequence of nucleotides. Before cell replication, DNA is going to be wrapped in a protective coating of chromatin that becomes the chromosome. So DNA is actually just in a chromosome. It's just been wrapped with the chromatin, and keep in mind that occurs during, just before, that there is going to be cell replication. If you have any questions, or think something was counted wrong, see me after class, circle it. We'll look it over, but overall, wasn't all that difficult in the exam. People done pretty well.
Was this one still out of 82?
Yep.
B is good.
There will be at the end a little bit of a curve.
I'm still confused about the difference between a chromatid and when it's called a chromosome.
You got a chromosome.
Yeah,
If you copy it, they stick together. They're called chromatids.
sister chromatids, right?
Whenever they come apart, they're chromosomes again.
Okay,
That's all it is. You start with chromosome, two chromosomes are stuck together called the chromatids.
okay.
And then when they're apart, they're,
So the X's, those aren't chromosomes. Those are sister chromatids.
well, the X is a copied chromosome. That is one of the bad things. The way they show this, it does not give people a true picture. That's a chromosome.
Okay.
You see them like that, they take the picture in metaphase. And when it's
Oh.
in metaphase, when it's in metaphase, it's not pulled apart, but they're going to the next phase, pull apart and make two cells.
So those are two replicated chromosomes that consist of two chromatids? Okay, got it.
They were chromatids when they were together, but the way they predict or show them is confusing.
So would they call that a duplicated chromosome? I think I've seen that before.
It's copied, yeah.
Okay.
Got it. I understand it now.
It's
It makes
one
so much more
chromosome
sense.
that's being copied.
Yeah.
And when they come together, they're held together by the centromere.
So in a cell that's not dividing, they're just chromatids, right?
You see them, they look like that. If you look at that, the ones that show as an X, they'll just look like that. Like almost like a worm, just string. And inside of that chromosome, then you've got the double helix strands. of
Okay.
DNA. That's all.
Okay, got it.
It makes it very confusing whenever you see some of these pictures and the way that they portray them. It's not really accurate.
Right.
And I still don't understand why they portray, I mean, I don't see the benefit in anything for doing that. But
Everyone followed.
Yeah, some guy just did it. Some guy was like, let's just show them when they're in metaphase. Here's a good idea.
Technically, it's two chromosomes.
Yeah, yeah.
It's a double. It's two chromosomes.
Okay, folks,
You can see double.
Maybe he was on drugs.
then we get into heredity. Something else that can be a little bit confusing.
We were attacked that.
Good thing is, the units that we've got left are not real long. In other words, we don't have that, a lot of information, so it really should be easier. One of the things you want to pay very much attention to is when we start doing the Punnett squares, you're going to have to do those on the exam. So make sure that we learn how to do these correctly. So this is Unit 14. Guys, once again, I'm not going to talk over you. If you want to talk with your partner, go outside. We started last time talking a little bit about traits passed down. And if those traits are passed down, what were they? In what form? That's what he was studying. don't worry too much about the history of this and like the blending hypotheses and so forth. Many of these theories they're talking about didn't come prove to be true. So this part is, which is not anything I don't think that you would have to study for an exam, but it's just saying there that there is something that parents pass to their offspring that decides what traits they have. What they call this a particulate. What we're calling it now are genes. One of the things that had these people questioning was, how come that is a great deal what Mendel talked about. Now remember, one of the things about Mendel, one thing that was important is he used pea plants. Pea plants, you can get a lot of generations quickly. It doesn't take, like in an animal, where it might take a year to have a new generation. Here, they could do it just, it was a monk in Austria, and his job was in gardening, and he became interested in that. Here is something else that you use these terms. Now notice, characters, the characteristic. What is the characteristic? He says, like flower color, that is a characteristic. What that characteristic actually is, the value of that characteristic, is called traits. Traits, each trait will have an allele. If the flower color is purple, there will be an allele that is purple for that trait. What it's asking. A characteristic is what a gene codes for. The trait is the value of that. If a gene codes for the characteristic of hair color, the trait would be brown, black, red, fluorescent pink, or whatever it is. But it does, you want to make sure that you understand how the term's used, because this gets tricky. And when I write a question, I have to write it in a way that is correct, precise, and you can't assume. You've got to know those definitions. This is telling you why he chose the pea plant and why it was so good. You can have many different characteristics. You can control the mating. Once you get into 1082, you'll find that a flower has pollen on the little stems called stamen. Up the center of the flower that will contain gold. Go to pollen the stamen on another plant that you want, and then just dust it around the top of the ovule, and it will become fertilized. What happens is that pollen actually goes down and crawls down in that tube, kind of makes a rope, and it goes to two different places within that egg. So all you got to do is get it on the top, and it will go down, the pollen. And plants are pretty easy to cross-pollinate. This actually shows up here. In nature, this is the carpel part, the part that carries the egg. And here you can show they're taking scissors and clipping these little things, which are the stamen that produces the pollen. Pollen is the sperm. Take a paintbrush, go to a plant that you want. All you have to do is put them on the end, and that end of that is sticky. That's where the nectar is at, going down into the flower. And that's the way pollination occurs with insects like bees. They go from one plant to the other. They will get that pollen, it'll get caught in their little ruffles. If you ever looked at them under a dissecting scope, the hair, and then when they go to land to get the nectar out of another plant, they knock off that pollen, and that's the way things fertilize. So they don't have to put it down in that little tube, just get it on the surface, and then that pollen will actually crawl down. True breeding plants. If Mendel was going to try to use logic to figure out what happened, he had to know what he was starting with. So what he did, he took two varieties of plants, purple flowers and white flowers. And he bred them and bred them and bred them like two purple flowers until the only thing that was produced for 13 generations was purple. And then he said, there's no other trait other than purple in that plant. He did the same thing for the white flowered plants, 13 generations. And that's called the true breeding plants. In other words, he just kept breeding them until the result were 100% purple or 100% white, depending on the plants. So then he would take the purple plant, true breeding, and the white plant, true breeding, and cross them. Anytime you take two things like that and cross those varieties over, they're called hybrids. This is a little bit different than in animals. In animals, hybrid can be like if you had a wolf and a coyote. We call those like the wolf dogs or the coy dogs, or coy wolves, whatever they are. But they are actually the same species. A wolf, a coyote, and a house dog are the same. There's no difference, same species. Now, if you take a horse and a donkey and cross them over, you get a mule. The mule is called a hybrid. It means it's got parts from both species, both parents. And there's a reason for that. When you get into genetics, you'll see. In plants, these are not necessarily talking about different species, but different strains of the species. And they're calling those hybrids. But most of these will normally be fertile. There are some hybrid plants that will not be if they're coming from two different species. So there's a difference between species and strain. The true breeding parents are called the P generation. That's parental. When you cross two pure breeding parents, the offspring will be called the F1, filarial one, filarial generation, F1. When F1 individuals self-pollinate or cross-pollinate with other F1s, you get an F2.
What did you say F represented?
Filarial.
Oh, Valerian. Thank you.
So Mendel took his true breeding purple and white flowers, cross them, and then plant and see, okay, what do we get? When he crossed these F1s with each other, many of the F2 plants, that's going to be the offspring of the F1s. Some had purple flowers, but some had white. All of the F1s were purple. 3 of them would be purple, 1 white. But you can just have one. That is called independent assortment, and nothing guides it. It's just giving you odds. It's like telling you 50 should come out heads, 50 should come out tails. Does it always work that way? No. It's just giving you a ratio and the odds. That's what we're doing here. So these at the top are our parental generation. whenever they are mated, all of their offspring had purple flowers only. These were the F1s, the first generation. When two of these were crossed, we get this ratio, 3-to-1. And notice, another big advantage of the flowers of using the peat plants is you can do a bunch of them at one time. So if you notice here, when he did that, he was doing it almost 1,000 times for offspring. So the more the results, more credible. Here are some of the traits that he checked for. On some, it occurred at the top, some midway through the plant. Color of the seed, yellow or green. Shape of the seed, round or wrinkled. Shape of the pod. Pod color. And were they tall or short? The main ones he was working with was going to be the flower color and the seed shape that we will see here, wrinkled or round. Now, one of the things, and I talked with Adriana the other day in the office about this, when you start to do these squares, you've got to use something to represent that trait that is understandable. Letters. You can use any, it doesn't matter, but you need to use something that you can tell the difference with. Think of a W. Hard to tell the difference between a uppercase W and a lowercase unless you really write them one bigger than the uppercase. On the other hand, take a B. It's easy to tell the difference between an uppercase B and a lowercase. So when you pick these things, try to pick something when you're doing it that can be distinguished. And whenever I give you one on the exam, you will be able to distinguish them. One thing that you need to remember that you'll be asked, all of the offspring of an F1 generally means it carries one allele for purple from one parent, one allele for white from the other. The trait that shows up is the dominant trait. The only way that we can tell if the trait's dominant or another is to test it and see which one shows up. That type of transfer is called true dominance. One trait will just totally dominate the other. Of the characteristic, maybe it's for purple color, or it can be a different allele for white color. And each of these genes are at a specific place on the chromosome, called a locus. This shows a chromosome. Now right there shows you what a chromosome, the basic shape will truly be. You can see it's not X'd. It's not metaphase. This is what they look like in the working version every day. Each one of these represents a chromosome from one parent. The allele for the flower colors, notice, will be at the same spot. It won't be one down here or whatever. It's going to be in the same area. That is the locus. When you have two chromosomes that are identical but just have different alleles, they're called homologous.
So an allele is a gene?
Yes, it's a specific version of the gene. We can say a gene codes for a characteristic like flower color. An allele would be, is it purple or white? It would be called a purple allele or a white allele, but it is the gene. Yep.
What were you saying about I'm sorry, I think we were talking about the bands for the alleles, if they're in the same place, or if they code for different things, or if they code for the
They
same
are in
thing?
the same place. That place they are located is called the locus,
Right. Thank you.
plural loci.
Were you saying something like mono? I think you were talking about if it codes for the same thing maybe, and it's in the same location and has a specific name.
It's an allele.
Right. Okay. Thank you.
Remember, an allele tells you what a gene just says it codes for a particular trait. The allele tells you what the specific trait that gene codes for.
Okay.
And remember, and these are simple little things, but sometimes if you don't think about them, it can be unclear. For each character, or character, in other words, for the flower color, its choices either have purple or white. Now, it's going to get one from each parent. They both could be purple, they both could be white, or one could be purple and one could be white. There's nothing to tell you what that combination is going to be, but there are two versions, two possible things that can show up. And think about this, Mendel did this without even knowing there was chromosomes or DNA. They were either in Italy or someplace in Europe, but we're talking about in the 1500s. We had no formal schools. What you did is you went to work with an apprentice, and you learned everything from them. Just one individual would be doing his work, and you'd stay there for maybe 10 years. This guy, I think he was 17 years old at the time. Knowing the math that they knew in the 1500s, constant, a mathematical constant that's about that long, Mendel said there has to be two things. Shows up, sometimes they don't. The one that shows up is dominant over the other one. It won't let that other one show. That's where we come up with dominant or recessive. And that's what we're talking about here. The one that shows up, the trait that actually you can look at and see, is called the dominant allele. The one that is there but does not show up is said to be the recessive allele. This is in the case of true dominance. Everything is not true dominance, as we will see. In the flower color, the example in F1 plants had purple flowers because they had an allele that was dominant, purple, but they still carried the white allele. Law of segregation, as we said, there is nothing that is going to determine what alleles shows up. It's all by chance, it's random. You've got two alleles for a trait. Let's say they're both different. You're only going to have one show up. You don't know which one it's going to be. Nothing determines it. And this is where we're getting into Punnett squares. Notice it says a capital letter represents a dominant allele, lowercase letter represents recessive. When you start getting into genetics, you'll find most of the time the gene will be given a letter like I. You may have seen that in some cases with the blood typing you've done. You can have 6 or more little subscripts at the top to show particular traits. It gets incredibly complex, and you have to have a legend right there to figure out what's what. We're keeping it simple, but just keep in mind, it gets much more. You can figure out what these ratios are with the Punnett square. Look at the top. We started with true breeding parents, so we know that purple is dominant, so it gets the 2 big P's. We know white is recessive to purple, so it gets a small case P's. Now there could be a situation where you mix two traits where white could be dominant over another color. We don't know. It depends on each individual setup as to where, which one shows up. We know all of them showed up to be purple, so we know that purple is dominant. We also know the only thing that white could give is the recessive trait. The only thing purple could give is the dominant trait. So we know that all of the flowers in F1 generation will be heterozygote. In other words, they will have two different alleles, a dominant and a recessive. So when you write what alleles that are present, that is called the genotype. Those are what alleles are there. In this case, a dominant and a recessive. That is called a heterodifferent zygote. If both were the same, you would say a homozygous, the same, homozygous dominant, or homozygous recessive is the way that you would refer to these. The way you write down what genes, what alleles are present is called genotype. When you look at something and see which one of these shows up, that is the phenotype. So the genotype for this plant flower right here would be heterozygote, PP. The phenotype would be what?
Purple.
Purple. That's what shows up, what it looks like. If we cross two of these, first you've got to set up the Punnett square. Normally, the most common method is female up and down, male across the top. It doesn't have to be that way. They work out the same, but when you have other people looking at these, if they know where you're putting things in the correct place, it makes it a lot easier, less confusing, but it will still work out. So we know they're all these two alleles, so we put one allele for this row, one allele for that row, one allele for that row, one allele for there, and then just combine. PP, big P, small p, small p, big P, small p, small p. Normally, and this is not true always, I wish it were, but sometimes, almost always, when you're combining these, put the dominant trait first. You don't have to. If you'd written it, this right here is a small case P and a big P, it would still be correct, but yeah.
What if you have, so I've had this case before, if you have like a 4x4 Punnett square and you've got two different alleles, so you have like a T and a W, and the T is recessive, but the W
Why would
is
you have a T and a
dominant.
W?
For, what was it?
Dad.
Two different alleles. So, I mean, for two different traits.
Okay, that is a dihybrid cross.
Right.
You're not gonna have 4 squares, you're gonna have 16.
Oh, right, I'm sorry, I meant 4x4. So,
We're gonna do one in a minute.
okay, but my question was, if I have a recessive T, basically, you have to always put the dominant in front of the lowercase, even if it's out of alphabetical order?
Alphabetical order has nothing to do with anything.
Okay, so just the dominant in front.
You'll get to see. It doesn't matter here if the small case P was in front, it makes no difference. Notice here, you've got one parent or one offspring that is homozygous dominant. It only carries the dominant trait. You've got 1, 2 here that are heterozygote. They carry both the dominant and recessive, but they're still purple. For a recessive trait to show and to be expressed, both alleles must have that recessive trait. Must be homozygous recessive in order for that to show up. Anytime, remember, we cross two F1s, the ratio will be always 3-to-1 dominant trait. And that doesn't, I mean, this applies to anything else, not just flowers, animals, whatever. But I will also tell you very seldom do you see a situation where one trait is by itself. There are other little areas on the gene called modifiers, restrictors, multipliers. Very seldom does one gene control a trait. There's usually quite a few. This tells you homozygous and heterozygous, as I've just talked about. This tells you phenotype and genotype, as we've just talked about. Notice this is the genotype. Phenotype is what you see, what shows up. A test cross is just taking two individuals, crossing them, and seeing what do you get. That is called a test cross. Notice it says, how can we tell the genotype of an individual with a dominant phenotype? Such an individual could be either homozygous or heterozygous. They both show up the same. Do a test cross. Breed the mystery individual with a homozygous recessive individual, and then if any offspring display the recessive phenotype, the mystery plant is heterozygous. This shows the true breeding parents, or not true breeding parents, but it shows two parents. If you cross these, we get everything's heterozygous. Okay, if we cross those two, what if we do cross one of these with the plant just like the parent plant? That was homozygous recessive.
Everything will be recessive.
So then we get two heterozygotes that are purple, and we get two this time that are white. 50-50 ratio. When you're asked on this, on the exam or whatever, to give the ratio, it's usually like 2-to-1, 1-to-1. You can say 50% or 100%. Either way. When you are only dealing with one characteristic, it's called a monohybrid. When you are dealing with two characteristics that are involved, it is called a dihybrid. Dihybrids, right here. That's what you're going to be doing. You're going to be doing one of these on the exam. So notice, at the top, this is a true breeding parent for yellow color and round seeds. Y represents yellow color, R the round seed. Dominant. The recessive version is to have wrinkled seeds, which will be the small case Y, and green color, which will be the small case R. The question that throws most people, everything comes down to one thing, the setup. If you get the setup correct, it's easy. Notice what we get here. If you take this Y and multiply it by that Y, and that Y, and multiply it by that R, we start to get setups. That's an experiment that they're doing right there. Notice here, this has got two traits, right? The Y and the R. This has got the Y and the R. That's all the parent can get. So if you multiply this Y times that Y, you get that. If you multiply that R by that R, you get that. That's the parent. That's what you get out of F1. They're all going to look like that in F1. Because both parents are homozygous. then you can set them up. I'll show you how to do that next time. It is not difficult, but we will walk through this. See you next time.
So those would be the F2 generator.